All right, this is part two of the senses. We're going to get into the special senses now. The first one is the sense of smell or olfaction. And the type of receptors are chemoreceptors. And they're concentrated in what's called the olfactory epithelium. Olfactory epithelium. So if you remember, epithelium is just a tissue that covers the outside surfaces or the inside surfaces of an organ. So this is an epithelium that's located on the upper, I should say superior nasal cavity. And so what happens is when you inhale a smell, which is basically a chemical, they're called odorants. They will stimulate the neurons in the epithelium. And then transmit an electrical signal to two olfactory bulbs. These are just neural tissue enlargements. And they're on either side of the crista galli, which is the on the ethmoid bone. We had the cribriform plate with the crista galli like right in the middle. So the olfactory bulbs are on either side of that. So, the crista galli. And then up to the brain. Where they are perceived as a smell. Okay. The next special sense is taste. These are also chemoreceptors. They're specifically known as gustatory cells. And on your tongue, you've got about 10,000 papillae. So if we were to take your tongue and cut it and look at it sideways, we would see these papillae. So up here, this is the surface of the tongue up here, and that's a papilla. And along the surface here and here are the taste buds. So let me just write that in red. And the gustatory cells, can't really see that very well. They are in the taste buds there. These are located in taste buds, found in the papillae of the tongue. So there are 5 primary taste sensations. One is sweet, sour, salty, bitter, and you may have heard of this one, umami. Which gives food kind of like a savory taste, or meaty. I've heard it described as. Some people also recognize alkaline and metallic as tastes, and then there's some people who believe that there's a water taste sensation as well. Okay. Oh, one thing to note, spicy is not one of those. Spicy? Nope. Okay, next one, hearing and equilibrium or balance. And the type of receptors that are found in this sense, or used by this sense, are mechanoreceptors. Hair cells. And they are found in hearing. They're found in the organ of Corti. It's also known as the spiral organ. And in equilibrium, they are found in the utricle, saccule, and the semicircular canals. Okay, the ear has got 3 parts to it. You have the outer ear, so ear parts or regions. You have the outer ear that includes the external ear flap, which is probably what you think of when you think of the ear. This is also known as the auricle. And the ear canal. And at the end of the ear canal, middle ear, is the tympanic membrane. Also known as the eardrum. And then you have the 3 auditory ossicles. the malleus, incus, and stapes. And the auditory tube that leads down to the pharynx. We were talking about this in class the other day. So the middle ear is like a little pocket of air that when you go on an airplane, the pressure changes and it's a little bit painful. So if you yawn, it opens up the auditory tube to the pharynx, which allows the air in that middle ear to equalize with the air on the outside of the body as well. This is also known as the eustachian tube. Okay. Okay, so we did the outer and middle. The inner ear is sort of the business end, where the stimulus is interpreted or sensed and then sent to the brain. So the inner ear has got, so for following sort of the path of sound, it starts at the oval window. And there's a region called the vestibule, and then the saccule and utricle. Actually, I lied. This is not the pathway of sound. This is just parts of the inner ear. The semicircular canals. And the cochlea with the organ of Corti inside of that. Organs of Corti. Okay, so the vestibule is kind of this larger region, and then the cochlea, this is a really not great drawing, but here's the cochlea. And oh boy. And you should really look at a diagram in the book because this is not very good. Here's the vestibule. And then the oval window is going to be, let's say there. The semicircular canals. And then you've got the saccule and utricle. Okay, so the cochlea is for hearing, and then all this down here is for equilibrium. Okay. So let's just focus on this bit for right now. Inside, let me go back here. So inside the cochlea, and all through here too, there is a set of tubes and they basically run up into all into here. And they're filled with a fluid called endolymph. The outside, basically, of the tube here is another type of fluid called perilymph. Okay, so you've got basically a tube within a cavity, and they have different types of fluid that fill the cavity and the tube that runs through the center of the cavity. The sound vibrate. I don't think I can fit this. Nope, not gonna be able to do that. The sound vibrations basically, they enter the ear, go through the ear canal, and then they hit the tympanic membrane. So first they're going to hit the tympanic. Oh wait, I can't spell tympanic for some reason. Tympanic membrane. And then those are in turn going to cause the auditory ossicles to vibrate. Which are in turn going to push on the oval window and then send the vibrations through the perilymph. Okay, so here's the oval window again. Basically, the bones are going to push on that, and then the sound vibrations are going to travel through the perilymph all the way up here, and then come back down the other side down that way. So they go up one side, and then they flip around up here and come back down the other way. So if we were to take that and cut it, we'd see that there's a set of 3 kind of tubes. Okay, so here's the inner tube with the endolymph is the blue, and then the 2 tubes here are filled. I'm sorry, perilymph. I'm sorry, endolymph. Jeez Louise. It's been a long freaking day. I literally have spent all day in front of my computer, and I'm tired. So here again is perilymph and the endolymph. And that's perilymph too. Okay, so the vibrations are coming up one side of the perilymph, and they're going to go up around the tip of the snail shell, up around there, hit the snail shell, and then turn back around and go back down the other side. Meanwhile, the vibrations are going to go from the perilymph and start vibrating the endolymph, and that's where the organ of Corti is there. Endolymph. And then they stimulate. So basically what's happening is you're vibrating this, and there are hair cells here that are being stimulated by the vibrations. And then there's a nerve that runs out through here. Signal is then sent to the brain via cranial nerve number 8, the vestibulocochlear nerve. One thing to kind of point out here, the vibrations are moving all through these kind of tubes basically, but the hair cells that are down here toward like the base of the cochlea They detect high frequency sounds, and then as the vibrations move up towards this, basically the tip up here, these are where low frequency sounds are detected, more towards the tip there. Okay. I'm already telling them to run out of time. Just gonna keep on going. Hearing loss can be caused by two things. What's called conductive, and this is when there's a problem transferring the sound waves associated with the transfer of the sound waves anywhere from the outer ear to the tympanic membrane and the auditory ossicles. So basically anything that's going to be vibrating. So this can be due just to like a earwax buildup, or through there can be fluid in the middle ear. You can get bacterial infections or damage to the auditory ossicles, or you can actually puncture the tympanic membrane, which is why they say be very careful. In fact, you shouldn't use Q-tips at all. Damage to auditory ossicles or the tympanic membrane. So that's conductive hearing loss. The other one is called sensorineural. And this is when there's damage to the inner ear. Damage in the inner ear, like the cochlea or the nerves. And this can be caused from some sort of inflammation of the nerves or an infection. So, and there are two tests that are really simple simple to use, but a little bit more complicated to interpret, that you can do pretty quickly to tell the difference. To tell the difference between conductive versus sensorineural hearing loss. There are two really simple tests that you can do to figure that out. And like I said, interpreting them is a little bit more complicated. Versus sensorineural hearing loss. One's called the Weber. Not like me. I got 2 B's. And the other one's called the Pinay. Oh, I'm sorry, Renee, not Pinay. Renee test. And you have to do them together. I mean, they're, like I said, they're really easy, but you take a tuning fork that's 512 hertz. You hit it, and so it starts vibrating. And with the Weber test, you hold the tuning fork kind of at the, well, it's, tuning fork somewhere along the top of the head. So there's the tuning fork. So that it's contacting the skull, right? So the vibrations are basically traveling through the bone down to the ear. If you have normal hearing, you're going to hear, or the patient, I guess, is going to hear the sound equally on both sides. If you have conductive hearing loss, then it's gonna be heard in the damaged ear. The one with hearing loss, oddly enough. And if you have sensorineural hearing loss, it's heard best in the non-damaged ear. Okay. In the Renee, remember the mastoid process is back here, kind of behind the ear. You can actually feel it. And here's the ear itself. First, you're going to hold the tuning fork up against the mastoid process so that it vibrates through the bone there. Hold the tuning fork at the mastoid process until the person says they can't hear it anymore, and then you take it off and then you hold it in front or next to the ear. So first there and then there. To the ear. Okay, so here it's vibrating through bone on the mastoid process, and then when they can't hear it, then you move it in front of the ear, and then you're actually hearing it through the ear canal, through air, basically. This is where it gets a little complicated. If they can hear it 2 times longer, then you record how much time they can hear it over here, and then how much time they can hear it over here. If they can hear it twice as long at the ear than at the bone, then they have normal hearing. If they have conductive hearing loss, it's heard longer at the bone than in air. And if you have sensorineural, then it's heard longer at the ear, but not twice as long like if in normal hearing, than on the bone. Okay. And if you want, you can look up, I found like a pretty good YouTube video explaining if you get a positive on this one and a positive on this one and a negative on this one, there's all these like different combinations between these 2 tests, and you can kind of narrow it down and figure out if the person has conductive hearing loss or sensorineural hearing loss. Okay, I'm going to stop there and then pick up with equilibrium on the next video.